At the end of our last lecture, we had just finished talking about the forebrain. So today we're going to start by talking about the midbrain and the hindbrain, then we'll move into our peripheral nervous system, and then at the very end we'll talk a little bit about development and change within the nervous system, sort of to wrap everything up here. So starting with the midbrain, we're going to look at the same image for actually both the midbrain and the hindbrain because they both conveniently show up together. So we have the midbrain highlighted here in blue, and this is mainly important because it contains structures that have secondary roles in vision, hearing, and movement. So our superior colliculi, these ones up here, they help guide eye movement and are important for gaze fixation. Our inferior colliculi, these two here, they help with sound localization. Not pictured on the image, but sort of on the inside, we have our substantia nigra, which projects out to the basal ganglia, and it helps us integrate movements. And the last thing that's kind of important here is, actually, if I scroll, oh, there we go, I had moved it somewhere. I have the fourth of our ventricles actually comes down through the brainstem, so it's kind of important to note there. Those are the important parts of the midbrain for us, at least. But now we're going to move on to the hindbrain. And as I said, we're going to be looking at the same image. So this time we're just going to be focusing down lower on the pink parts. And when we talk about the hindbrain, we can talk about the pons, the reticular formation, medulla, and cerebellum. The reticular formation is one of those that goes between the midbrain and the hindbrain, which is why it's not pictured here. It is important in regulating sleep and arousal, attention, and things to do with motor activity. So that's the main roles for that one. The pons in the middle here is also related to sleep and arousal, and that's because it connects with that reticular formation. So there's some overlap in functionality there. We also have the medulla at the lowest part down here. And this helps us maintain sort of those essential life processes, things like having our heartbeat and keeping up respiration and things like that. So the medulla takes care of those basic life functions. And the last part is actually sort of a weird one. So the cerebellum is one of those things that nobody seems to agree on where it belongs. Some people group it in with the hindbrain, some people group it in with, I've seen it with the midbrain. I've seen it on its own as sort of a separate, not grouped thing. For those who are curious, the cerebellum is sort of this very visually distinctive portion that sits at sort of the back and the base of our skull. So it looks very distinct, and the name of it actually, cerebellum, means tiny brain, because it looks like its own standalone, smaller version of the brain. But wherever we decide to group that cerebellum, that's mostly going to be involved in refining movements, things that are initiated by our motor cortex, and then it's going to sort of moderate them and make sure that the speed and intensity and direction and everything all work together so that we can maintain our balance. So it helps with precise movements and also seems to play a role in motor learning. So learning to do actions as we do them over and over again. As I've said, that's sort of a very intense crash course on all of those different parts of the brain. I wanted to point out that there's a really nice table in the textbook, table 3.2, and it summarizes all of the major structures of the brain and their functions, so that might be a good thing to have handy just so that you can reference it easily. Now, in our process to sort of finish up the central nervous system, we're going to talk about the spinal cord because that is how we link our peripheral nervous system to our central nervous system. So we're going to talk briefly about the spinal cord and then move through the peripheral nervous system stuff. For our official definition of the spinal cord, we can talk about it being a cable of neurons that helps carry commands from brain to muscles, as well as from brain to organs, and it takes all of our sensory information and brings it up to the brain. So it helps route information into the brain and out of the brain to and from the rest of the body. So if we flip to the next slide to see some of the visuals here, we have sort of a outer view and then a cross-sectional view of what our spinal cord looks like. So we have our vertebrae with these, well, sort of a cross-section of the spinal cord there, and we can see that it has white matter on the outside and gray matter on the inside. We also know that these the nerves that connect to our body and then up to the brain exit the spinal cord at different points. So we have our cervical nerves, our thoracic nerves, lumbar nerves, sacral nerves. These all sort of project out of the spinal cord at different levels. The higher up on the body it is going to cover. So the cervical vertebrae connect to our head and our neck, our shoulders and our elbows. Our thoracic vertebrae end up connecting to the lower part of our arms, our upper torso, our internal organs. The lumbar nerves that connects to our intestines, legs, and feet. And then the sacral nerves at the bottom here, they control our hips and buttocks. So lots and lots of different functions, and they're all divided out nice and evenly. We can also talk about where those nerves are exiting. So at each of those levels, we can have both a dorsal and a ventral root. So if you remember back to our sort of directionality of those terms, dorsal meaning towards the back and ventral meaning towards the front. So we're saying that the dorsal, so on the back, that's where the sensory neurons are entering the spinal cord, and the ventral towards the front is where the motor neurons come out of the spinal cord. So it's nice and easily organized with a pattern there. And we can talk about one more thing that our spine is really good for, and these are spinal reflexes. So a reflex is a simple automatic movement that occurs in response to sensory stimuli. So the usual example that I use is if you accidentally touch a hot surface. So your hand will immediately pull back, and it happens so quickly that you're not even aware that you've done it until it's already happened. So you touch a boiling hot pot or something, and a sensory neuron from your hand is going to send a signal up through the body, and it's going to connect to your spine. There's a little bit of a weird shift in sort of zoom levels, I guess, here. And so along the dorsal root, that's where it's going to enter into the spine. Once it hits the spine, that signal is going to sort of branch into two. So part of that signal is going to head up to the brain to let your brain know that your hand hurts. But because touching something really hot can cause damage over a long period of time, we want a quick withdrawal. And that's where these interneurons happen. So an interneuron is going to sort of make a snap decision at the level of the spine as opposed to sort of at the level of the brain, and it's going to cause a muscle reflex where the muscle immediately contracts and pulls your hand back from the thing that was causing you pain. So this decision happens before that signal has reached the brain and you've been able to make a conscious decision, because we've cut out sort of all those extra steps of routing up and back down again. So instead of having to travel all that distance, it just has to travel the distance of that interneuron, and then it goes straight back into the motor neuron and contracts the muscle. So that's why our reflexes can happen so swiftly. And the last thing that I wanted to talk about in the topic of the central nervous system is how we protect the central nervous system. Since our central nervous system is so it's responsible for so many different things, we definitely want to do what we can to protect it. It's vulnerable to damage that might come from things like physical trauma or incoming chemicals or things like that. So we have what are called the meninges, which are, it's a protective 3-layered membrane. So if you've heard of like the dura relating to the brain, that's the dura layer. That's the outermost of those 3 layers. It's sort of a tough outer coating. Then we have the arachnoid layer, which helps separate our blood and our cerebrospinal fluid. And then we have the pia layer, which is the innermost layer. And this sort of wraps around our central nervous system, and it allows our central nervous system to float in cerebrospinal fluid. And I think we already mentioned that spinal fluid as sort of a cushion, I guess, that the central nervous system can exist in, and it helps protect against, say, sudden movements or being jostled around. We also really care about our blood-brain barrier because it helps limit passage between the bloodstream and the brain. So if stuff is in our bloodstream and would theoretically have access to the brain, the blood-brain barrier keeps it in the bloodstream without allowing it into the brain unless it meets certain criteria. So it would keep out things like toxins or extra neurotransmitters or things that we don't need in our brain, and only allows things that are supposed to be in. And this happens because the capillaries in our brain are very tightly joined together. So there are only tiny, tiny gaps, as opposed to where in the rest of our body, the capillaries are very spread out and allow things to pass through fairly freely. So this is a special piece that's evolved to help protect our brain from other stuff in the body. And that is our whirlwind tour of the central nervous system. Next, we're going to talk about the peripheral nervous system, and you can kind of breathe a sigh of relief because our central nervous system was an awful lot, but the peripheral nervous system is a lot more simplistic. We're going to look at sort of 2 divisions, and then we're going to divide one of those divisions in 2 as well, and that's kind of it. So our peripheral nervous system is going to be fairly mundane, I guess, in comparison. One thing I do want to mention is sort of another term that we can use to talk about things moving into and out of the central nervous system and the peripheral nervous system. So if we talk about efferent, I'm going to pronounce them like overly exaggerated here. So afferent, if we talk about like an afferent neuron, then it's heading towards the central nervous system. So that's usually going to be things like sensory neurons. If we have efferent, with an E, it's moving away from the central nervous system. So that would be things like motor neurons. So these are just other terms that you might see. So it's a good idea to be aware of these terms. Okay, so if we're going to talk about the peripheral nervous system, let's define what we're talking about. And this is basically all of our nerves that lie outside of the central nervous system. So it's everything that we haven't talked about already. So we have 2 different categories of nerves that we can talk about. We already kind of mentioned our spinal nerves, so the ones that are coming out of the spine. Once they've left the spine, that's when we start thinking about the peripheral nervous system. So there are the ones that leave the spine and they sort of join and disconnect from the spine at each of the vertebrae. Whereas we also have cranial nerves, and these nerves enter and leave at the underside of the brain. So these ones actually skip the spinal cord and go straight into the brain without going through that spinal cord. And the ones that typically do this are ones that are doing functions that are very, very close to the brain already. So that's why they're called cranial nerves, because they tend to be sort of in and around our craniums or our heads. This is sort of the list of all of the different cranial nerves. We don't care too much about each of these right now. We'll come back to them as they're important. Mostly what we're going to care about is that we have sort of facial cranial nerves. So these are the ones that control the muscles in our face. We have our olfactory and optic cranial nerves, and these bring in sensory information from our eyes and our nose. We also have sensory information from our sort of our inner ear, so the vestibular cochlear, because it's our vestibular system and it's getting information from those cochlear canals in our ears about what our orientation is and if we're moving or not. But as I said, we can go into more detail on these later on in the course as they become important, but this is just giving you an overview of some of the things that our cranial nerves do. What we're going to care a lot more about are those divisions that I just mentioned. So the first of our divisions here is to talk about the somatic nervous system and the autonomic nervous system. So our somatic nervous system is our voluntary muscle and sensory receptors. So the things that are bringing in sensory information from our body, and then also the motor neurons that are going out to allow us to manually control each of our actions. So the things that are involved in us touching and feeling things, but also then moving our hands to interact with those things, that would be our somatic nervous system. The other side of that division is our autonomic nervous system. And this is what controls automatic involuntary functions. So I always remember this as the autonomic system is automatic. So the same pre-word chart. And it is our autonomic system that ends up being divided further into our sympathetic and parasympathetic systems. So if I hop, too far. If I hop back here, we can simplify it and talk about the sympathetic as our go response. It's usually activating things. This is what's involved in our fight or flight response. So when you're faced with a threat and your body sort of revs up to either fight off that threat or run away from that threat, that's your sympathetic nervous system. So this is going to increase our pulse and increase our respiration. It's going to give the body the resources it needs to move. It's going to change our blood pressure so that things can move move blood and those important resources to all those important internal organs and our brain. All of those things are involved in this go response within the sympathetic nervous system. And then the parasympathetic nervous system is the opposite of that. Instead of a go response, this is a stop. So this is going to slow the activity of most of our organs. It's going to help us sort of slow things down. And for those who are more visual, we have this handy little chart here that shows our sympathetic, the revving up, and our parasympathetic, the calming down, and they tend to work in opposition of one another. So most of our organs have connections to both of these systems. So when we're faced with a threat, we might dilate our pupils because we want to see more information, gather more information, and know how to handle the threat. When the threat's over and we return to normal, the parasympathetic nervous system would constrict those pupils, bring them back to normal. And sort of back and forth here where they act in opposition to one another. And maintaining a balance between these two systems is what keeps us at our sort of homeostasis point, where our body is happy at an equilibrium. So we're going to have sort of a back and forth between these to keep at our happy midpoint. And that is it for the peripheral nervous system. I promised that it would be a short one, and I delivered. Now we're going to move on to our last couple of slides talking about development and change in the nervous system. And I don't know about you guys, but I thought that this was one of the most interesting reads, at least so far in the textbook, because the development part was, development, but talking about different ways of sort of repairing damage and ways that people have tried to get around damage, I found it very interesting. So it'll be kind of neat to run through this stuff with you. I just want to draw your attention to the fact that this is only a couple of pages in a textbook, and it's trying to summarize lots and lots of research and lots of literature on these topics. So again, it's a lot more simplistic than what is reality. Just, again, be aware that if this is something you're interested in, you can absolutely take entire courses on the development of our nervous system and how our nervous system can change and respond to sort of damage and recover from damage and stuff like that. But we're going to start at the very beginning, which are the basic stages of development for our central nervous system. So our nervous system begins as a hollow tube that eventually becomes our brain and the spinal cord. So it's going to look something kind of like this. And again, we're going to keep the details fairly low. I have taken sort of developmental biology classes, and we could spend an entire course on this. But what we care about is the fact that this tube here is sort of forming as it grows. So we'll notice that towards the end, it's still open so it can extend. And then towards the middle, it's closed up and sealed up. So we care about here our neural groove, so this little dent in this outer surface, and the neural fold, which are where the raised edges on the outside are. And then the neural plate is that bottom part, and that is all over our notochord, which is sort of the preemptive stages of our spinal cord and things like that. So as we get further down, as things start closing in, these are basically coming up and wrapping around. So they're growing to enclose that tube in the center. And that's kind of related to things like we were talking about our starts with a V, ventricles. There we go. that would be sort of these tubes. That's how they would originate. So we don't care too much about this stage here. What we're going to focus on instead are the 4 distinct stages of the development of the nervous system. So we're going to look at cell proliferation, migration, circuit formation, and circuit pruning. So let's start here, looking at proliferation and migration first. This is one of those rare times where I find the names of these things actually match very nicely with what's happening. So during proliferation, these cells are multiplying prolifically. They're making lots and lots of divisions, and they multiply and divide at a rate of up to 250,000 new cells every minute. So this is a massive growth, of massive multiplication in the number of cells that we have. So this happens in that ventricular zone, the area that surrounds the hollow tube that we just talked about forming. So it's surrounding what will become our ventricles and the central canal towards the end. So proliferation is where we have rapid multiplication or rapid division of our cells. So the next part is migration, which, as the name might imply, is when those cells start moving to their final locations. So these newly formed neurons move from that ventricular zone that they proliferated in to the final location that they should be heading towards. So most of them are going to be moving perpendicular from the ventricular surface. And what this means is if we have this is the surface of that tube, they're going to be moving this way. And it seems kind of weird. How does a cell migrate straight away from where it was created? Well, that's where our radial glial cells come in. So these guys here. And they're going to guide migrating neurons. So basically what they do is they turn into scaffolding that our neurons can then climb to head in the right direction. So the radial glial cells help make sure that they get to the place that they need to go. Now, the really cool part is that as these neurons move, as they migrate away from where they were created, they will start sort of specializing and picking up different functional roles. And the role that they adopt will depend on when it's existing, when it was created, when it arrives at those locations, and which location it arrives at. So it seems like the environment that they arrive into determines what they do. And this means that in early life, at least, we have a lot of flexibility with our neurons because as they arrive in different areas, they can take on different roles. And this is what makes it's kind of like stem cells and things that haven't specialized yet. At this early stage, especially in, say, fetal brains, then you can move tissue from any part of the brain to any other part of the brain, and it'll just sort of pick up whatever traits it needs to work in that new area. So if neurons end up in the wrong place, then it's not so much of a big deal because they use what's around them to figure out what they should be doing. And we also have some mechanisms to get rid of errors and such, which we'll talk about just in a second here. The next stage we want to talk about is circuit formation. And this is when we form connections or make circuits of our neurons. So the axons of those developing neurons start growing towards the places that they need to connect to. And this is a really cool process. So these sort of proto-neurons will develop growth cones, which are basically little protrusions at the end of their axons, and they'll sample the environment looking for directional cues. So they're going to look for things like chemical or molecular signposts. So maybe they're supposed to head towards this particular chemical, or maybe they're going to head away from that particular chemical. And by sampling the environment and migrating using those chemical signals that are coming from other cells, they can sort of wind their way from where they've started to where they need to go. So this is how our axons can start connecting at different places all across the brain. And during this process, we end up making a lot more connections than we actually need. So that's where the last step comes in, and this is circuit pruning. This is when we start getting rid of the excess neurons and synapses. So the theory here is that the process of having these axons migrate to the place they're supposed to connect, it's fairly fraught with error. Axons can take a wrong turn, they can connect to the wrong places, sometimes too many of them end up in the right place. And so what happens is that things aren't optimized. So when we're very, very young, we have lots and lots and lots of connections. But we start pruning or getting rid of those connections that aren't effective. So if they aren't functional, or if they're in the wrong place, if they're not doing a job, we can get rid of those connections. And that's what the pruning is here. There are sort of two categories of circuit pruning that we can talk about. The first is if the neurons don't connect where they're supposed to, if they miss a target cell, if there are already active neurons there that have taken up that role, if they're too late getting to that location, they'll be pruned, they'll die off. We also have sort of a more refining type pruning, where the brain sort of determines which neurons are active and which ones are actively being used, so which ones are firing along with those around them, and those that don't fire when they should, according to patterns of firing, those would be pruned as well. And again, I'm sure you can imagine this is a very complex topic that we could spend tons of time on, and may talk about more later on in the class. And then we can talk about some general terms. We can talk about plasticity, which is the ability of our synapses to be modified. So if we talk about plasticity in the brain, then we're saying that the brain can form new connections as we experience new things. So as we form new memories, the plasticity in our brain allows us to make new connections and learn those new things. So I kind of mentioned how we start off with a lot of neurons, and then they can make connections, and then we prune them, and we end up with sort of less neurons over time. So as humans age, we tend to lose neurons, and in the first few years of life, we lose them fairly dramatically. But also, as we get older, despite having fewer neurons total, we end up having more and more connections. So fewer neurons, but more synapses between those that remain. So that's part of that plasticity with age. And we might also even see something like reorganization, where we have an overall shift in connections within our brain. So we might reorganize after an injury. Or we can see things like those who are born blind. They've never had to use the visual center of their brain because they don't see. So reorganization would allow them to sort of take over that part of their brain and use it for something else. So there's some evidence that those who read Braille, the part of their brain that would have been their visual center, ends up being more responsive to sort of sensations and touch sensations. We also find that they have a much larger increase in the area of the brain in their motor cortex, not their motor cortex, in their somatosensory cortex, the incoming signals, for their index finger, which is what they use to read Braille, ends up getting a lot larger. And while our ability for this reorganization is a lot higher when we're younger, so we are far more plastic when we're until about the age of 2 or 3, and we can recover from a lot of stuff when we're especially young, as we get older, we can still have fairly dramatic shifts in how things connect. And the textbook talks about one example of those who are born with syndactyly, which is basically webbed fingers. So they have all of their fingers connected by extra skin that never got pruned during development. And if they undergo surgery as an adult to separate their fingers, their brain will adjust and recognize distinct independent finger representations in as short as 7 days after the procedure. So even as adults, we can still have this dramatic and rapid reorganization. And again, we don't know everything there is to know about how this all works, we're mostly stuck relying on things like case studies or experimental techniques in non-human animals, but it's a very cutting edge field and they're learning more and more every day. And logically, this discussion of how our brains can change and how they can adapt and learn new things and connect in new ways definitely brings us to this idea of, well, what happens after damage? How do we see recovery after damage in the brain? And though there are lots and lots of ways that we can suffer damage to our central nervous system, the two most common are stroke and traumatic brain injury. So in a stroke, this is caused by loss of blood flow to parts of the brain. So this can be either ischemic or hemorrhagic. So if it's ischemic, it would usually be caused by the blockage of an artery, so usually a blood clot or some other kind of obstruction. Hemorrhagic strokes are a lot rarer, but occur if an artery ruptures and sort of blood spills out. And the part that I found really interesting here is that it's not actually the deprivation of resources. It's not the loss of oxygen or the loss of glucose in the neurons that cause the most damage. That's sort of a secondary cause of damage. But most of the damage that actually comes from, exocytosis. So as some of the neurons die, they end up releasing glutamate. And that flood of glutamate ends up triggering other neurons to be overstimulated and then die. So it's sort of this chain reaction where when one starts having problems, it can actually trigger others to have problems, and it's sort of this ripple effect. So that's the primary source of damage. And then there's also the issue with oxygen and glucose deprivation. So not a great thing to happen. The second of these, the traumatic brain injury, or TBI, is a condition that's caused by some kind of external mechanical force, sudden acceleration or deceleration or penetration that physically damages the brain. So a big hot button topic nowadays is talking about concussions, which would be a sudden acceleration or deceleration where the brain suffers trauma. So even if a concussion doesn't lead to loss of consciousness, it can cause sort of long-term problems, and especially if you have repeating concussions, if you have multiple concussions successively, it can cause long-term brain damage that can start causing things like amnesia or memory loss or attention deficits. So it's a big problem for especially in sports. So, if those are the two most common ways that the brain can suffer damage, how do we potentially recover from this kind of damage? There are two major things that we need to look at here. The first is regeneration, where severed axons can regrow. This does occur occasionally in mammals, especially in the peripheral nervous system. Inside the central nervous system, it's a lot more complex. But in the peripheral nervous system, if we had, say, a broken arm or something, and it severed the nerves that were in are connecting to our fingers, if we had received treatment quickly enough and the cell body itself wasn't harmed, even if the axon died off because it was severed in the break, that axon would regrow. It would actually use the existing myelin sheath as sort of a guide to sort of slip back where it needs to be and pick up where it left off. But that doesn't always work. It's very difficult for those axons to find the correct path, especially if there are other signals around. So in the central nervous system, there are those specific chemical signals that got our axons to the right place at the right time by following those chemical trails. But once we're done growing, those signals go away. And so it's almost impossible for those axons to navigate back to where they were because they don't have the roadmap to follow anymore. So Their pathway can either not be findable because they can't navigate anymore. It might also be blocked by other cells that are in the area. There might be axon growth inhibitors, which are problematic. So there's lots of stuff that can go on and block that kind of regeneration. So it doesn't necessarily happen as often as we would hope it would. The other option is neurogenesis, and this is when new neurons can be born. And this is another one of those sort of rare events. It does still occur fairly commonly in some specific areas of human brains. Specifically, we've seen it. I think this was actually experimentally in just sort of mammals in general, not human specific. But we tend to see new neurons being developed in the hippocampus and in the area around the olfactory bulb. So the hippocampus, that makes sense because our hippocampus is involved in forming new memories. So new neurons means new connections, means learning new things, kind of makes sense. However, there's a little bit of debate on whether the stuff around the olfactory bulb makes sense. Some people have proposed that it's like, oh, well, you're forming new memories that associate smells with those memories, but that's another of those where they haven't quite replicated the studies, so they can't agree on whether that's for sure or not. But the general consensus seems to be that the hippocampus is one of those for sure sites of neurogenesis in adult brains. So if regeneration and neurogenesis aren't really common, especially in humans, then what else can happen? What else can help us recover? And here we can focus on compensation and reorganization. We already kind of talked about reorganization where different parts of the brain can take over. So if there's a part of the brain that you're not using, it can pick up extra functions. Or if one part of the brain is used a lot and other parts are used less, then that part that's used more will take up more area. And so this is also part of maybe compensation, where not so much that you're just sort of shifting the proportions. In compensation, we're saying that there's an injured tissue that's going to lose its functionality, and then uninjured tissue will take over the function of those that can no longer function. So maybe you have a head injury and it causes you to lose control of a certain part of your brain, but you still need that functionality that was controlled through that part of your brain. So other areas can start picking up some of the slack. They can start using or performing some of those operations and allow you to start recovering. If we lose just a couple of neurons, then maybe other neurons in the area will start adapting their functionality. So you might have presynaptic neurons growing more terminals so that they can send extra signals to neighboring neurons that weren't damaged. And maybe postsynaptic neurons are going to add more receptors to receive signals from more other areas, so they can compensate around what was lost. And we actually had a little bit of a discussion about this on Tuesday, where we were talking about what happens if somebody, say, lost their hemisphere or had part of their brain removed. And this sort of compensation and reorganization can allow people to have other parts of their brains, or maybe the other hemisphere, try and help step up to fill in that problem. So if I think we mentioned the fact that our left hemisphere tends to be specialized more towards language because that's where our Broca's and Wernicke's areas are. But if the left side of the brain is damaged, that person can eventually recover their speech, especially if they still had some functionality in the right hemisphere and they weren't sort of completely lateralized. So some people can recover just fine, especially if the right part of their brain was already okay with helping out with language. That part of the brain could take over or compensate for what was lost on the left. Now, we do typically see that people will have some kind of recovery, but it's not necessarily going to be a 100% recovery. when it comes to recovery of sort of the spinal cord, especially, you might start thinking about stem cells. So these are undifferentiated cells, cells that don't have sort of a taken on role yet, that can develop into specialized cells. So they could turn into neurons or muscle or blood cells or whatever. So this typical thing that is used would be embryonic stem cells. So harvested from embryos, they would be pluripotent, which basically means that they can differentiate into any cell in the body. And they do that based on what chemical signals are in the environment around them. So the same way that when I talked about our neurons migrating to the correct area, they sort of know what their role should be based on what's around them. Those signals can still tell these stem cells what they should be doing based on what's around them. We have adult stem cells as well, but they're less potent. So they're going to basically be confined to areas with high replacement, things like our skin or our intestines. They're also really common in our bone marrow. And so they're kind of already set in what they should specialize into, whereas those pluripotent embryonic stem cells, they can turn into anything. And that's why there's so much push for research with embryonic stem cells. And again, that's going to be one of those big things in the future that people continue to try and research because there's so much potential for improvement. And so that kind of ties us nicely into our sort of wrap up for the chapter, which is basically to say that we know a lot. We've learned a lot about the brain. We know a lot about how it functions, but we definitely don't know everything. There are still so many unanswered questions. There's still so much that we don't know, but people are starting to do lots and lots of research. They're starting to push forward to understand how all of these things work together. And so I always say it's going to be an interesting couple of decades as this stuff continues to move forward. This is definitely one of those fields that's going to evolve very, very quickly, but at least we now have a solid foundation on the basics, and we can keep building on that throughout the rest of the course.